Bruce is a graduate of Davis, California. He got his DVM degree there. And then he went to the National Zoo, and he was a resident under Dr. Dick Montali for 2 years. And then he went back to Davis and obtained a PhD. And he's been at San Diego Zoo, I guess, about 2 years now. 3 years now, sorry. Well, he is a staff pathologist. And Bruce is going to discuss with us this next hour the pathology of marsupials. Bruce.
First thing I'd like to do is thank the C.L. Davis Foundation and Dr. Thompson and Loyola University for putting this on and giving me the opportunity to come. I'd also like to thank Dick Montali for letting me go over the marsupial pathology records for the National Zoo and for him providing a lot of the slides that I'm going to be showing you. Dr. Bob Renate contributed a couple of slides, and my new boss, Dr. Lowenstein, made this nice title slide for me and contributed several of the other slides I'm going to be showing. Thanks, boss. First thing I'd like to do is go over just a little bit of taxonomy. Since marsupials comprise a pretty large and diverse group, it's kind of hard to keep them all straight if you don't understand at least a little bit of taxonomy. Fortunately, it's pretty easy. The major division is based on dentition, and so the first group I'll discuss is the Diprotodons. They have 2 incisors in the mandible. They're basically all herbivorous animals. The phalangerids are things like cuscuses and brush-tailed possums. Pteroids are the gliders. Macropods, of course, are kangaroos and wallabies and pademelons and tree kangaroos and things like that. Protoroids are things like rat kangaroos and batons and whatnot. Phascolarctidae is one and only koala, and there's 3 species of wombats. This is just to show you what the dentition looks like. This is a ventral view of a wallaby mandible showing the 2 large incisors in the mandible, making them diprotodons. This is a representative of the phalangerids. This is a female spotted cuscus. This is a pteroid. This is a lesser sugar glider. And of course, the macropods are represented by kangaroos. This is a southern yellow-footed rock wallaby. The arboreal macropods like the tree kangaroos. This is a baton. And of course, for those zoos that can afford the very best, this is the koala. What Dick likes to call the poor man's tree kangaroo. And this is naked-nosed wombat. The other major group is the Polyphotodons. They have at least 4 incisors in the mandible. They're mostly carnivores or omnivores. Didelphids, of course, includes like the North American opossum. And for whatever it's worth, the North American and South American versions are called opossums, while the Australian ones are just possums. So don't ask me why, but that's the way it is. The Dasyuridae is a large and diverse family. I'll show you some representatives of them. Paramelidae are like the bandicoots, and I'm not going to talk about this group at all today. Mostly what I'm going to be going over are the macropods, koalas, and some of the dasyurids as far as diseases go, but I'll show you some of the representatives of these groups. This, of course, is the North American opossum. The dasyurids are, as I said, a large and diverse group and are represented by everything from small marsupial mice like these fat-tailed dunnerts to the dibbler, koaris, qualls. This is a spot-tailed quall, and the Tasmanian devil. This is bandicoot. I also want to go over a little bit of marsupial anatomy and physiology, since they have a lot of interesting features. Some of the things that have been proposed as defining features of marsupials, of course, is the pouch, which is where the term marsupial comes from. The pouch is the marsupium. Marsupials were thought to lack a placenta, and so some people have suggested that as the major defining feature of marsupials. Others have proposed that the fact that the ureters develop medial to the genital ducts is the most important feature, and I'll show you what the anatomical significance of that is in a few moments. And there's actually been a proposal that the fact that marsupials lack brown fat as the major distinction between marsupials and eutherian mammals, but traditionally the pouch is the main distinguishing feature. Unfortunately, it's not necessarily the best feature, since not all marsupials have pouches. Some of the smaller marsupial mice don't, and some species only have a pouch during lactation. Also, not everything that has a pouch is a marsupial. The echidna also has a pouch. As far as the time that the neonate is in the pouch, there is a period in which the neonate is permanently attached to the teeth, and that period of time corresponds to the period of gestation in eutherian mammals. And as a general rule, the pouch opens forward in Diprotodons and rearward in the Polyphotodons, but there are some exceptions to that. This is an example of a koala pouch, and as you can see, if there's nobody in there, it's not a very prominent feature. There's just a small opening right here. And if you open that up, you can see that the nipples are fairly small, or the teats are fairly small, and they are an exception to the rule I just gave you, in that their pouch tends to open a little more rearward. In the Polyphotodons, they often have multiple offspring. This is a North American possum with 7 neonates attached to the nipple, whereas the Diprotodons generally will have a single fetus in the pouch. When the teats are being used, they tend to get extremely long, and the mammary glands get well developed, and the pouch continues to enlarge until, as you can see, as the animals, as the neonates get pretty old, the pouch is a large structure and they can hardly fit into it anymore. As I said, some people have claimed that the lack of placentas in marsupials is an important defining feature, but I think by modern definitions, most people would agree that marsupials actually have a placenta. The main difference is the allantois plays a very minor part in marsupial placentation. In fact, they have the smallest known vascular allantois. Chorioallantoic fusion does occur in phalangerids and macropods, but chorioallantoic placentation occurs only in bandicoots. The rest have a choriovitelline or yolk sac placenta. The female reproductive tract, most of you are probably aware that there are paired lateral vaginas which meet a central vaginal vestibule. There are 2 cervices and 2 separate uteruses. And the reason for this structure is thought to be due to the fact that, as I said a few moments ago, the ureters embryologically develop medial to the genital ducts. And as a result, at least the embryologists claim that if the genital ducts were to fuse and form one vagina, it would obliterate the ureters. So that is supposedly the explanation for why they have this unusual reproductive tract. At parturition, a birth canal forms in the connective tissue between the median vagina and the urogenital sinus. And that birth canal is transient in most marsupials, but in macropods, once that canal is formed and becomes epithelialized, it's a permanent structure. And this is an example of the female reproductive tract. Down here is the urogenital sinus. The lateral vaginas come off and meet at the median vagina here. If we were to open this up, there would be 2 cervices in here, and then 2 separate uteruses and 2 ovaries. This is where the birth canal forms, and since this is from an adult wallaby, there is a permanent birth canal in here, but in other species, this is just connective tissue, and the birth canal reforms at every parturition. There are some unusual features of the male reproductive tract also. The scrotum is positioned cranial to the location of penis, which is kind of unusual. Wombats don't have an obvious scrotum. The testes only descend during the breeding season. And the penis usually has a prominent fork at the end, except in like kangaroos and things. This is kind of a dark slide, but it's the only one I have that kind of gives an illustration of this arrangement. The penis is back here and the scrotum is way up here, so kind of an unusual backward arrangement, but it seems to work, I guess. This is the penis of a North American possum, showing this branch here. The, I guess, obvious reason for having this is so that they can inseminate both uteruses, but it's worth noting that the urethra ends right before the bifurcation, but sperm can flow down two grooves that run out to the tips of these processes so that they can inseminate both uteruses in the female. This is a glider, sugar glider, that's being electroejaculated, and you can see how much longer the processes are on that one. As far as secondary sex glands, the marsupial males have a prostate and often very prominent bulbourethral glands, but they lack seminal vesicles. As far as the musculoskeletal system goes, the epipubes are the marsupial bones, which most of you have probably seen. They're present in all living marsupials. They were lacking apparently in the Thylacine, which is now extinct. Syndactyly is fusion of the second and third digits of the rear feet. It occurs in all diprotodonts and also in bandicoots. It's thought to function as a grooming organ. Most marsupials, except for the bandicoot, lack an osseous patella, and for clinicians, growth plates remain open much longer in marsupials than in eutherians, so that can be a nice thing to know when you're evaluating radiographs. This is the hind foot of a koala, just showing the syndactyly. There's fusion here of the second and third digits. Even in the larger macropods with these really large feet, they have the same kind of syndactyly. Here's the foot of a wallaby showing the fusion of the second and third digits. These are the knees of a koala, and just showing the lack of an osseous patella. This can cause some confusion because there is a fibrocartilaginous pad there that you can palpate. It feels like a patella. It can cause problems if it luxates. This actually was, these radiographs are from a koala that was having problems with luxating one of its patellas, and the clinicians were very frustrated when they found that they couldn't find them on the x-rays. As far as the teeth goes, the main things to remember are that marsupials generally lack deciduous teeth except for the posterior premolars. And interestingly, in the diprotodonts, mainly the koalas or the kangaroos and wallabies, the molars move forward along the jaw and are eventually lost as they grow older. This animal has some problems with the dentition here, but you can see a tooth that I think is about to be lost here as it's moving forward. Cardiovascular system, the main thing to remember is that marsupials lack foraminal valley in the heart like eutherian mammals do. They just have a series of fenestrations, kind of like a bird heart. And the other main thing to remember is that the heart is often about 30% larger than eutherians. The main thing I want you to remember for gastrointestinal system is the macropod stomach has three main chambers: the sacciform region, tubiform, and the hind stomach. This is an example of the esophagus is coming in here. These large sacculations are the sacciform stomach. The tubiform stomach sort of runs alongside it and both and down here in the hind stomach. This is one of these stomachs opened up. There's usually a squamous portion right around the cardia of the stomach here, but this is the tubiform region, sacciform region here, and the hind stomach here. For whatever it's worth, the macropods are able to undergo proteolytic digestion in all the chambers of the stomach, at least the youngsters are, so that you don't have problems like you do in ruminants. If you get milk or formula into one of these other chambers of the stomach, it doesn't ferment and cause problems there. They can digest it no matter where it ends up. As far as lymphoid and hematopoietic systems, the important thing to remember is that there is a cervical thymus in all diprotodonts that I know of. And there's only a cervical thymus in koalas and wombats. And that's an important thing to know, as I'll show a little bit later. Some interesting gene whiz things. Only IgG and IgM have been identified so far in marsupials, and there seems to be a lack of immune suppressive mechanisms associated with pregnancy in marsupial females. From a clinical standpoint, it's worth knowing that unlike eutherian mammals in which immunoglobulin absorption only occurs in the youngsters in the first 24 to 48 hours or so, it continues throughout pouch life in marsupials. So they're constantly getting those immunoglobulins from the milk as long as they're in the pouch nursing. Persistent Splenichymatophoresis occurs in some marsupials, especially the smaller basiurids and things like that, and it can be so spectacular as to cause confusion over whether or not it's hematopoietic neoplasia. This is the neck region of a koala. The head's up here. This is thoracic inlet down here. And this is the cervical thymus that I was telling you about. The other lobe I kind of nicked and it's reflected back there. But this is really important because you can look all you want in the thorax and you're not going to find a thymus, and it's important to evaluate the thymus in koalas. Another gee whizzer, there's no corpus callosum in the brains of marsupials, and as I mentioned earlier, they lack brown fat. This is the brain of a koala section sagittally, and there's no corpus callosum in there. Okay, let's get into diseases now. The diseases of macropods, these are the main big ones that you'll find written up in the literature and which have been seen at both National and San Diego zoos. I'm going to talk about a lot of these, so I won't go into them right now, but some of the ones that we don't see so much in captivity are things like the herpes virus infections. There is marsupial herpes virus that affects primarily the diprotodonts. It has been reported in some polyphotodonts, but causes fatal infections, lesions are in mucocutaneous areas, respiratory tract, GI tract, basically systemic infections. There's widespread seropositivity in the wild, but I can't find any reports of actual known deaths in the wild from the herpes virus. It mainly seems to be a problem when they're brought into captivity and stress or something activates the virus and causes severe systemic infection. I don't have any pictures of tetanus because it's not very photogenic, but macropods are highly susceptible to tetanus and should be vaccinated for it. There's also macropod pox virus, which I'm not aware has been reported in any zoo collections, but has been seen in the wild. Just causes generally skin lesions and is not generally fatal. Tisner's disease has been reported. I'm not going to get into a lot of the parasites, although some of them are interesting. I'll get into coccidiosis a little bit later, but remember that strongyloidiasis is a gastric parasitism in macropods, not intestinal, and Marsupial strongylus is the lungworm that has a very wide host range, affects an awful lot of marsupials. First disease I want to talk about is lumpy jaw. Hard for me to tell if that's in sharp focus, but the pathogenesis of this is still a little bit controversial depending on what factors you think are most important in contributing to it. Most people, I think, would agree that feeding coarse hay and grasses with sharp awns and that sort of thing contributes to it, because that coarse plant material can get stuck in the gingival sulcus there, set up a nidus of infection, causes necrosis in the mandible, and periosteal osteoproliferation around the nidus of infection. Some people, though, think that the movement of the molars along the jaw, when abnormalities develop during that progressive movement of the teeth, that contributes to it also. This is a mandible that's been sectioned. This is a tooth here, large pocket of necrosis down underneath the alveolus here, and bony proliferation around it. Now, in Australia, there have been some good studies that would indicate that the primary pathogen involved here is Fusobacterium necrophorum, and actinomyces and some of the other organisms that are commonly associated with this are only secondary invaders, although most of the literature from the U.S. would indicate that actinomyces is the primary pathogen. But I don't have time to get into the reasons why, but I think the Australians have some good evidence that it's Fusobacterium necrophorum is the main pathogen. It doesn't only affect the mandible. This is an animal with some early lesions in the maxilla. Can spread to the hard palate. There's a fistula here, oronasal fistula in the hard palate, large area of necrosis on the hard palate here. Can also spread to involve the base of the tongue. This is a ventral view of the tongue of a wallaby that had lumpy jaw, large area of necrosis and inflammation there. And many of these animals will develop lung lesions as well. This is a really severe pyogranulomatous pneumonia in a wallaby that had lumpy jaw, and actinomyces was grown from this lung. Salmonellosis is also a big problem in captivity and in the wild. Just an incredible variety of Salmonella serotypes have been isolated from macropods, and since so many of them can carry the organism asymptomatically, it's sometimes worth doing fecal screenings for it. Can cause enteric infections as well as septicemias. This was from a young wallaby that was being hand-reared in our children's zoo at San Diego, and it actually seemed to be doing okay at the time, but the clinicians were doing routine fecal screenings for Salmonella, and it came up fecal positive, and so was euthanized, and it was found to have this interstitial ammonia at necropsy, from which we isolated Salmonella enteritidis in pure culture. Dermatophytosis can also be a problem in captivity and in the wild. Both Trichophyton and Microsporum can be involved, and this is a wallaby with some pretty good ringworm lesions. Esophageal Candidiasis is also important, especially in young hand-reared animals. Partly, I think, because sometimes the young ones, at least in our experience at San Diego, don't respond well sometimes to artificial milk formulations. They'll get diarrheas and things like that, get put on antibiotics, and eventually will develop candidiasis as a result. But adults also get it. This is esophagus from an adult wallaby, or red kangaroo, I'm sorry, with candidiasis. Histologically, there's no challenge. It's just lots of Candida pseudohyphae yeasts. Toxoplasmosis is another big one. Most of you are probably aware of the problems associated with this in captive animals. It's worth knowing that there have been some good reports that would indicate that at least some of the cases in captivity are reactivation of latent infections. So if you have a case that's occurring, for example, in an animal you have in quarantine, which wouldn't have any exposure to cat feces or anything like that, doesn't mean it's not toxo. It could be activation of a latent infection. This is a brain from an animal with toxoplasmosis showing a small area of necrosis and inflammation. And you can generally pick up the tachyzoites in the lesion, so it's not too difficult of a diagnosis. Some of the other protozoa that we've had problems with at San Diego is an entamoeba species, which has caused gastritis and gastric ulcers. This is the lesions are generally localized for some reason right in the cardia of the stomach, right where the esophagus joins in. There's a perforating ulcer right there. And Lynn Greiner did the work on that, and they identified it as an entamoeba by immunofluorescence testing. We haven't seen much of the gastric amoebiasis for a while, but we did have an interesting case of a Protozoal Esophagitis in a Parma wallaby not too long ago. And it was kind of unusual in that this is esophageal mucosa up here. There's a large pocket here filled with these large ciliates, lots of inflammatory cells. And although I'm probably not going to be able to point them out right now, there's also amoeba in these lesions, which I think are the primary pathogens. This could be an amoeba down here. And I think the ciliates are just kind of coming along for the ride and forming this kind of spectacular columns of organisms on the esophageal surface. Coccidiosis is also a problem. This is an oocyst from a fecal. It's mainly a problem in weaned animals that are just being weaned, and in animals newly introduced into groups that already have coccidiosis endemic. But there are a number of species of Eimeria and Isospora that occur in macropods, and at least the Eimeria species can cause fatal hemorrhagic enteritis, so it's worth probably treating it if we find the oocyst there. Nutritional and exertional myopathies are also a well-known problem in these guys. We don't see it too much anymore as a nutritional problem, but because a lot of the large macropods are relatively sedentary in captivity, if there's anything that spooks them and causes them to run around, like a predator in the area or a clinician with a blowpipe or something like that, they'll really run themselves death, basically. This is an example of really severe exertional myopathy. One thing that you're not going to find much in the literature, but which most pathologists and clinicians at zoos will attest to as an important problem of mortality is self-trauma. A lot of the macropods are a little bit skittish, and it doesn't take much to cause them to jump into a fence. In fact, we had a problem just recently when the Wild Animal Park opened up a new exhibit in which they're going to allow the public to walk through the exhibit where there are wallabies and things running around loose. And the first experiment with this, some people were trying to corner one of the wallabies just for kicks, and this is not that animal, but it basically looked very similar. It jumped into a fence and broke its neck. It's worth knowing, though, that although this one has fairly spectacular hemorrhage all through here, and it's not a difficult diagnosis, in some cases you won't see that much hemorrhage around the head of the neck, but you may see really severe pulmonary edema. So if you see nothing else but pulmonary edema, you might want to think about the possibility of head trauma. Another thing you don't see very much in the literature, but which we've seen a fair number of cases of both at San Diego and at the National Zoo, is gastrointestinal accidents. This is intestinal volvulus, or mesenteric torsion, in a kangaroo from the National Zoo. And I don't have a good explanation for this, but we do have a fair number of cases in our collection and at National. We've also had some idiopathic cardiomyopathies and other cardiovascular problems in our macropod collection. This is from a Goodfellow's tree kangaroo. It had a cardiomyopathy. The heart is opened up here, and there's a large atrial thrombus right there. I haven't been able to come up with a good explanation for why these guys develop these cardiomyopathies. In the past, they used to have a lot of problems at San Diego with aortic mineralization. This is the aorta, and you can see the knobby surface is all due to mineralization. It was a real frequent finding back in Lynn Griner's days, and we don't see it too much now. Another thing that we used to see, or that was seen with some regularity at San Diego, was renal calculi. In the literature, there's only been a few reports of renal calculi in macropods, and those, I think, were uric acid and magnesium phosphate. But unfortunately, it seems like the ones that were seen in the early days at San Diego were never analyzed, so I don't know what kind of renal calculi we have there. I think we're going to do a carousel change, I hope. I'm sure Dr. Montali had already talked about the problems of mycobacteriosis in tree kangaroos, and this is an example of it. I wasn't here when he was going over his part. I was loading my slides, so for all I know, he showed this very same picture. But this is a case of the Mycobacterium Osteomyelitis. There's obvious infiltrative lesions in here. I'm not going to go over a lot on the mycobacteriosis. I think you guys have heard a lot about it already today. Except to say a couple of things. One is, as far as types of mycobacteria that can be involved, just let your imagination run wild. Anything you can think of has probably been isolated from a marsupial at some time or another. And the other thing is the lesions are not always granulomatous. They can even be suppurative. This is vertebral bodies from one of these tree kangaroos. I think you can see a pocket of suppurative exudate right there. And as I recall, although I didn't find this in my survey of the National Zoo's records, as I recall, when I was there as a resident, we actually had a case of a pyometra in a small marsupial that we isolated Mycobacterium smegmatis from. But that may have been a surgical case and not a necropsy case. But in any event, histologically it looks like what you would expect. There's a lot of bone destruction, fibrosis, granulomatous inflammation, and the multinucleated giant cells, if you do an acid-fast stain, will have organisms in there. Granulomatous lesions in the lung are also common with mycobacteriosis. And now, I think a great animal from a pathologist standpoint is the koala. They have a really interesting array of diseases. They're Diprotodonts, just like the kangaroos and wallabies and things, but their teeth are a little bit almost more rodent-like. They're not as large and prominent as they are in the wallabies. When they're born, they're obviously pretty small, about a half a gram to a gram at the most. And I'm sure most of you have probably seen those neat nature specials on TV where they show the newborn marsupials crawling through the hair and making their way to the pouch. But unfortunately, a certain percentage of them never make it all the way to the pouch. And this is an example of one of them. You usually find them as little autolyzed blobs in the bottom of the cage. If they happen to make it there, they're pretty cute by the time they're ready to leave the pouch. This one's, I think, about 7 or 8 months old. Some of the interesting anatomical features of koalas are that they have the longest cecum of any known mammal. The cecum is thought to function in the breakdown of tannin protein complexes. As you can probably imagine, eucalyptus leaves are not necessarily a high-quality diet. They're not that high in protein. And one of the problems that you have with utilizing it as a dietary source is that the tannins in the leaves will bind the proteins and make them unavailable. So one of the things that goes on in the cecum is that there are bacteria there that will break down these tannin protein complexes, and the bacteria then utilize the protein, and then when the bacteria turn over, the koala will utilize the bacteria as a protein source. So that little cycle there enables them to make the maximum use out of their protein sources in the leaves. It's also thought to be important in water balance. The material in there is not watery, but it's fairly moist, and because it's so large, there can actually be quite a bit of water stored up there. And koalas generally don't drink much water. They get most of their moisture just from the eucalyptus leaves. They also produce what are called pap feces, like some other hindgut fermenters. The mothers, when they're beginning to wean their offspring, will produce this soft fecal material that the young will eat, and it provides them with the fecal microflora to begin eating eucalyptus leaves. As I mentioned before, they have a paired cervical thymus. They have a prominent gastric gland, which I'll show you in a minute. And they progressively accumulate more and more brown granular pigment in their hepatocytes throughout life. And I'll show you some of these things in just a few minutes. As far as their brains go, only about 60% the size of a eutherian mammal brain for the same body weight, so not terribly intelligent animals, but I guess they have enough to get by. Their adrenal glands tend to be a little on the small side also, so you have to be aware of that, or you'll get thrown off by the small size. I also want to mention these hepatic pseudo-inclusions, which I have some pictures of in just a minute, that have caused some confusion in the past. Koalas have really neat hands. They have 2 digits opposing 3 that enable them to grip onto tree branches and stuff a little bit better, and this is an example of the hand in action. And this picture really doesn't do justice to the size of the koala cecum, but this is it here. This is the ileum coming up. Cecum is on this side, and the colon is on this side. And you may be able to see in both the cecum and the proximal colon, these white longitudinal lines correspond to folds in the mucosa that increase the surface area, and they're real prominent all through here. And let me tell you, when you open up this segment of the GI tract, the smell of eucalyptus oil would just about knock you on your you know what. Histologically, the cecum and the proximal colon have this really dense carpet of bacteria adherent to the mucosal surface, and this is that bacterial population that breaks down the tannin protein complexes, so that's a normal finding. This is the stomach, showing this large gastric gland on the lesser curvature right near the gastroesophageal junction. This slide is in upside down and backwards on purpose to keep the same orientation from the previous slide. The esophagus is coming in here. This is the gastric gland and the rest of the mucosa. The function of this is not well understood. It's thought that it may have something to do with getting the eucalyptus leaves ready for complete digestion, but basically this entire mucosa is a normal gastric mucosa that they have in addition to this gastric gland. Histologically, it almost looks a little bit like a bird proventriculus at low mag, but it's basically normal gastric mucosa inside these glands. Grossly, the koala liver is really dark, and in older animals can be almost black, as in this case, and that's due largely to this accumulation of brown pigment. You can see it occurs in hepatocytes, and there is at least a similar appearing pigment that occurs in Kupffer cells. But for some reason, the Kupffer cell pigment will stain positive for iron and lipofuscin, but this hepatic pigment doesn't stain for anything that anybody knows of. It's thought that it's probably related to residual breakdown products from the digestion of the eucalyptus leaves, but nobody's ever been able to get it to stain for anything. These are the pseudo-inclusions I was alluding to earlier in the hepatocytes. By electron microscopy, they appear to be glycogen, although I think there have been a few reports in the literature claiming that this is the marsupial herpes virus infection, but there's no lesions associated with it. That's the giveaway. This is an otherwise normal hepatocyte. These are leukemic cells in the sinusoids, which we find in an awful lot of our koalas. Going over the diseases, I'm going to talk about most of these things in showing you slides, but some of the things I don't have slides on, I'll talk about now. Chlamydia is a really big problem in the wild, and in fact is thought to be one of the big factors contributing to the decline in koala populations in the wild. There are two syndromes associated with chlamydiosis, a respiratory and ocular syndrome and a urogenital infection, and I'll talk in more detail about those in just a second. Mycobacterial infections are also seen, but it's mostly problems with skin infections in the wild. We don't see it too much in captivity for some reason. GI disease used to be more of a problem in captive animals in the past, but we don't see it too much now, and I don't know why. Toxoplasmosis has been reported, but because koalas are arboreal herbivores, they don't get exposed to it very much, so it's not a real significant disease problem. As far as chlamydiosis goes, there were originally 4 syndromes identified. I prefer to kind of lump them together as the ocular and respiratory forms and the renal and genital forms. But these syndromes were first identified in the late 1800s, but it wasn't until 1974 that it was found to be a chlamydial etiology. And interestingly, there are 2 separate strains. One causes the respiratory and ocular lesions, and one causes the urogenital lesions. And both of these strains are unique, and they're only found in koalas. They have never, at least so far, nobody has ever identified these strains of chlamydia in any other marsupial or other mammal. It can cause overt or subclinical disease, so you can be bringing healthy animals into your collection that are carrying it and shedding it, so you need to be aware of that. Transmission, nobody's really sure about the mode of transmission, but the obvious ones have all been suggested. Inhalation is probably important in the respiratory forms. Ingestion could also be a problem, and it's thought that possibly the females could be transmitting it to their offspring when the offspring eat cat feces. Venereal transmission is also possible for the urogenital forms, and flies may contribute to the spread of the ocular form. Diagnosis, all the obvious means: complement fixation, isolation of the organism, signs and lesions are generally what people rely on. Treatment's a problem because the same types of bacteria that are effective against the organism are also effective against the fecal microflora. And if you wipe that out, the animals tend to sort of waste away and die. But, excuse me. The disease can be controlled in captivity, even in very high-dense captive populations, by testing every 6 months every animal in your collection. Those that are positive are isolated for at least 20 months while they undergo treatment, and then you must keep separate husbandry for all the animals. As I said, we don't see this too much in captivity now, but at the time this picture was taken, they were having some clinical problems in the koalas at San Diego, which sound in retrospect like chlamydiosis, but at that time the etiology wasn't known. And these are kidneys showing some really severe nephritis, lots of scarring and infarcts in the kidneys. They did isolate Pseudomonas from this, but the pattern of disease that was going on at the time is kind of suggestive of a chlamydial disease, so that could be an example of it. Cystic ovaries have been reported a lot in koalas, but the literature is really frustrating because they describe these things as anything from cystadenomas to para-ovarian cysts to anything else you can imagine. And we don't have any slides that I could find in our files, any microscope slides that I could look at and try to get an idea of what these cystic lesions are. But some people attribute them to a sequelae of chlamydial infection, but others think it's more likely related to phytoestrogens in the eucalyptus leaves that they eat. In any event, it has been associated with infertility. Now this is the reason why I've been hammering home the point that koalas have a cervical thymus. This is, again, a ventral view of the neck of a koala. The head is up here. This is the end of the mandibles right here. And this is where the thymus is supposed to be, but it's about 20 times larger than it should be. And this, I think, is a thymic lymphoma in a koala. We've had at least 2 or 3 cases of this in the last few years. And interestingly, thymic lymphomas have never been reported in koalas, but they frequently report involvement of lymph nodes of the neck. And I'm suspicious, I haven't been able to confirm it yet, but I'm suspicious that at least some of those cases are actually thymic lymphomas, and the people that are making the diagnosis just weren't aware that the koala has their thymus in the neck there. Most of the animals that have lymphoma are also leukemic, at least in the terminal stages of disease. This is the buffy coat from an animal in the terminal stages of disease that had a white count of over 100,000. What's amazing is they'll survive for an awfully long time with a white count like that. But once they die or are euthanized, you find leukemic infiltrates everywhere. This is the eye. You can see all these leukemic infiltrates throughout. Pituitary, lots of infiltration there. This is gastric gland. I already showed you the liver a few minutes ago that had infiltrates in it. Replaces the medullary cords and the lymph nodes. And also, unfortunately, can affect the mammary gland in females that are raising offspring. This was a case of a female that had a 4-month-old joey in the pouch. And generally, it's very difficult to hand raise them successfully before about the age of 6 months, which is when they first start to leave the pouch. And this animal, they were trying to keep the mother alive as long as they could, at least for another 2 months, to give the offspring a chance to survive. But as they were doing routine weighings on the baby, they discovered that it was basically wasting away. And the reason is we found that, that the mammary gland was heavily infiltrated by the neoplastic process, and when we expressed some milk out and smeared it on a slide, it looked like a lymph node aspirate. It was just nothing but neoplastic cells, so I don't think the kid was getting much milk. It was mostly neoplastic cells. Another really interesting disease problem that koalas have has been called the craniofacial tumor of koalas in the literature. It basically resembles sort of an osteochondroma-like lesion. It generally affects the flat bones of the skull, but in this case, I think we had the same disease problem affecting the humerus, causing this real destructive lesion here. But the astute clinicians out there in the audience will also see that there is some fluffiness around this zygomatic bone right here that you don't see on this side. The treatment for this was to amputate it. Again, this was a female that had a joey in the pouch, and they were basically trying to keep her alive long enough to get the joey out and just taking parts off of her to keep the disease under control. This eventually progressed to the point where she was suffering severe exophthalmos, and they had to get a plastic surgeon in to remove that part of the zygomatic arch. But as you can see, some of the tumor was left behind. It continued to progress, formed a larger and larger lesion, causing more eye problems, and actually progressed to the point where she couldn't open and close her mouth very well, and she had to be euthanized. Histologically, there's a bit of variation in what these craniofacial tumors look like. This is a lesion from the hard palate. Epithelium is up here. Down here, it mostly looks like rather benign bony proliferation, although you may get a hint of some chondrocyte-like cells down there, to more aggressive-looking lesions with a lot of pleomorphic chondrocytes in areas of osteoid deposition. And the other real big one that we see a lot of in the recent past at San Diego is aplastic anemias. This is normal bone marrow from an adult animal, fairly moderate cellularity for femoral bone marrow. And this is from an animal with aplastic anemia. And this is, believe me, the most cellular area in this bone marrow. Everything else looks just like adipose tissue. And what you find clinically in these animals is they act perfectly normal, but they have really severe anemias and pancytopenias. And in fact, this animal had a PCV of 6 for over 2 weeks before it was finally euthanized. The clinicians got the curators to agree that if the PCV dropped below 5, they would euthanize the animal, or dropped below 6, they would euthanize the animal. But it was a difficult thing for the curators because the keepers would swear that there was nothing wrong with this animal. It would walk around and eat just like normal, but at any moment it'll just crash and die. This is a little tiny brain of the koala, but the problem here is cryptococcosis, and it basically follows the same kind of disease pattern you're probably familiar with. Nasal infections can develop, go through the cribriform plate, cause CNS infections. Histologically, looks like what you would expect. Here are the organisms here, thick capsule around them. Very little to no inflammatory response, at least in the brain. Pulmonary involvement is common. They get a little bit more inflammation, but mostly just lots of cryptococci in the lung. What's interesting is there have been some studies in the recent past on Cryptococcus neoformans, and it is known that there are 2 variants. Cryptococcus neoformans, variant neoformans, has 2 serotypes, A and D, which is the types we're most familiar with, causing disease in cats and AIDS patients, and the environmental source is thought to be pigeon droppings and things like that. The other variant, Gatti, has 2 serotypes, serotype C and D. And until recently, the environmental source of this organism was not known. It does not appear to be associated with bird droppings. But a recent study in Australia found that the organism can be isolated in the air, around flowering, a certain species of flowering eucalyptus tree. This is Eucalyptus calmodulensis. And throughout the year, you can isolate the organism from the bark and the leaf litter underneath the trees. And it turns out that this is one of the favorite food sources of the koala. And the serotypes that we see in our koalas is serotype C, which is associated with this eucalyptus tree. The only thing we haven't been able to do is isolate the organism from our Eucalyptus calmodulensis on San Diego Zoo grounds, which is where we get our leaves from. But at least at this point, the epidemiological evidence from Australia is kind of convincing in that they trace the pattern of distribution of these trees in Australia, and it fairly closely correlated with the distribution of type C serotype infections in humans and marsupials in Australia. So kind of an interesting thing to keep in mind. Another big problem in koalas is pneumonias, and Bordetella bronchoseptica is probably the most commonly isolated organism, but Pseudomonas and all kinds of other things can be isolated. The last thing I'll mention just briefly, we don't see it much in our collection, but there has been identified what's called a koala stress syndrome. Nobody's quite sure what causes it, but for unexplained reasons, animals will just go off beat, start wasting away, and die. Some of them will have gastric ulcers, as this animal did, but it's basically just an unexplained wasting disease. Now, for the really astute pathologists out there and clinicians too, there's a pretty obvious pattern of disease that that we've been seeing here in the koalas, when you've got things like high prevalence of lymphomas and leukemias, aplastic anemias, wasting disease, cryptococcosis, osteochondroma-like lesions. It all sort of fits the pattern of what used to be seen in cats with FELD in catteries. And in fact, Mike Worley, the virologist at the San San Diego Zoo has isolated a retrovirus from these koalas, and just about every animal in our collection except, I think at last testing, we may have 2 animals that are negative by PCR for this virus, but at least Mike and I are convinced that most of the disease problems we see in our collection are associated with this retrovirus. Mike has tested a lot of animals from the wild, and there's also a very high prevalence in the wild, and interestingly, there's a very high prevalence of lymphomas in the wild in koalas that may correlate with the distribution of the virus. So there's a lot more work that needs to be done on that, but Mike's doing a lot of interesting things. I don't have a lot of time to go over diseases of polyphotodons. A lot of these are the same things you would see in macropods, with a few exceptions. Post-mating mortality in antechinus species I'm going to discuss in just a second. The rest of these, as I said, are fairly similar to what you would see in a macropod, except that neoplastic diseases in the polyphotodons, there are some predilections to lymphoid tumors and smaller dasyurids. There's a genetic predisposition of lymphomas in the fat-tailed antechinus, which is a little marsupial mouse. And benign and malignant mammary, hepatic, and adrenal proliferations are fairly common. And as I mentioned before, Splenichymatophoresis is frequently confused with, or can be confused with neoplasia in these guys. Now, this post-mating male mortality is a real interesting phenomenon. It occurs in these genuses, but mainly in antechinus, which is a little marsupial mouse. Oops. This is one of these guys here. And the interesting thing is, the males will breed their first year of life, and virtually all of them will die about 1 to 2 weeks after mating. In one study, 12% of the males survived the first breeding season, but that was, the measurements taken like a month after the breeding season, there were still 12% alive. It's not known how many of those were alive for the next breeding season. But what's interesting is you can take these animals out of the wild either before or after they mate, and the males will still die in captivity and the females will survive. Most of the males are severely anemic immediately prior to death, and in general, the cause of death is attributed to severe stress. They have GI ulcers, marked lymphopenia, neutrophilia on a hemogram, adrenal hypertrophy. In some cases, there can be listeriosis outbreaks or other kinds of sort of opportunistic infections. They have problems with babesia. You'll see cytomegalovirus inclusions in the prostate, and there's also irreversible spermatic failure. Even if you can get them to survive the first breeding season, they stop producing sperm for some reason. Nobody has a very good explanation for why this occurs. It occurs in all antechinus populations that have been examined. Some of these others, it may occur in only certain populations or only in certain years. It may happen one year and not in another. So it's kind of a very interesting disease problem or pattern of mortality that nobody has a good explanation for. This is the obligatory mycobacteriosis slide in a tiger claw. One thing that I find interesting is that it's not only the Australian marsupials that have problems with high prevalence of lymphomas. This is a short bare-tailed opossum from the National Zoo, which is a South American marsupial, and the National Zoo has a reasonably high prevalence of lymphomas in that group, so kind of interesting. Some of the other tumors they get, this is a mesothelioma in Koala, nodules on the liver and on the serosa of the intestine. Adenocarcinomas, the mammary gland, have also been seen fairly frequently in things like tiger claws. And I have to say something about North American possums. And as you guys probably know, probably the only significant cause of mortality in possums is getting run over by cars. And if you open up any of those roadkills, you'll see Physoloptera in the stomach. And that's about the extent of my knowledge of possum pathology. And that's it. Anybody have any questions? the only, I think, good evidence that we have that there's a connection between the retrovirus and the diseases is just the type of diseases, and the pattern is very suggestive. We've had problems doing studies of like immune suppression in koalas because we don't have any reagents at this point that recognize T and B lymphocytes and all of that sort of thing. Mike has had persistent problems with the virus because there aren't any marsupial cell lines that he can infect with the virus, and so he can only keep it in short-term lymphocyte cultures from koalas. So he's been working on cloning it, but for any of you that have worked on cloning of viruses, it's sort of a laborious task, and since Mike is working alone and has other things he's involved in, progress hasn't been as fast as we would like, but we're hoping in the next year or two to write some grants and get some more funding and some more help to look into it, because I think one of the things that we need to do is try to find some negative koalas and compare the pattern of disease in them to the ones that have the virus, and also find the ones that have the virus and look for signs of clinical immunosuppression. Everybody's hungry. What's that?